This is a production of Cornell University.
All right, but we're talking about plants, and then let's talk a little bit more about how the plants actually get these nutrients into their system. Okay, foliar uptake is, unless we're spraying it, it's not a big deal, not necessarily a big source of nutrients, except unless you're an aquatic plant. Most plants are basically getting their nutrients through root uptake. And basically you're looking at a cell wall here, and with a cell membrane, okay, inside that. And basically what the plants need to do is to get those nutrients from out in the soil into the cell. Okay? Now they have a lot of different ways of doing that. We're going to talk about that. But the membrane basically functions to concentrate these nutrient ions inside that cell versus outside. Okay, now how they do that is primarily through respiration. They're using respiration to get energy. They use that energy then to pump and discriminate those ions inside their cells. Okay? Now the result of this is basically we have an uptake of nutrients that are sensitive to biologically limiting things. Plants, cells, and the roots are basically aerobic. They're not photosynthesizing. Okay, they're aerobic. They need to have oxygen to respire so they can do this. If the system gets cold or there is a lack of oxygen, you're going to limit biological activity, and as a result, you're going to limit this nutrient uptake. How these work, we've talked about this earlier with issues of proton pumps, but basically we're looking at, here's the root tip. The plant cells must maintain a charge balance between the inside of the cell and the outside of the cell. Okay. Now the inside of the cells generally has a higher concentration of nutrients, but they still need to maintain this charge balance. So if they are bringing in cations, positively charged ions, they need to counteract that positive charge. And they can do it two ways. One is they can bring in negatively charged ions, anions. Okay? Certainly you can imagine if they had to pump in potassium, they could pump in a nitrate, for example. Potassium is the positive charge, nitrate is the negative charge. Okay. They also can pump things out. If they're bringing in positively charged ions, they can pump out protons to counteract that change of positive in, positive out. And this is where we get in the term of the proton pumps. Now certainly you can also imagine that depending upon what their sources of materials are, they could potentially be bringing in anions. And nitrate is a really good example. If nitrogen is their primary source of nitrate is their primary source of nitrogen, it's a negatively charged ion. So they're bringing in negatively charged ions into the system. So in essence, they have to either pump out negatively charged things or bring in positively charged things at the same time. Now, why is this important? Think about fertilizing. If we fertilize our plants with ammonium versus nitrate, two N sources, if we're bringing in protons, so think about the rhizosphere. Okay? If the plant is bringing in ammonium as its source of nitrogen, it's going to be pumping out positively charged protons. What is that going to be doing to the rhizosphere pH? If I'm increasing the concentration of protons, I'm going to be lowering the pH, right? Now, spin that. What happens if the plant, not me, if the plant is bringing nitrogen in as nitrate? Not ammonium. Nitrate is coming in as a negative charge. Now, it could be bringing in positive charges at the same time to maintain that charge balance, or it could be pumping out negative charged ions. What is it going to be doing to the rhizosphere when it's bringing in nitrate as its nutrient source of N? What's going to happen to the pH of the rhizosphere? It's going to go up. So depending upon what we're fertilizing, we in essence are going to be changing or playing with that pH. And what does pH do to the availability of nutrients? Changes them. If I start lowering the pH, I'm going to start making micronutrients more available off of 6.5. If I start raising the pH, I'm going to start making macronutrients more available as opposed to 6.5. Does that make sense? Okay, so how the plants are feeding also has a huge impact on the environment that they're in and the availability of nutrients. Now, we're talking about this, the plant uptake of those nutrients. Okay? But it's important to think about this in a sense, you know, if we're interested in maintaining fertility for our plants, we can't just think about the uptake of the plants, of those nutrients. We need to think about what's in the soil solution and how it's interacting with those reserve areas. Okay? Now, if that's what we're talking about, we're really talking about release mechanisms. We're talking about release mechanisms of nutrients, or cations or anions, from this reserve area into the soil solution where it can then go up to the plant. Those release mechanisms are basically threefold. There's sort of three controlling issues here. If it's a cation exchange reaction, cation exchange capacity, it's basically charge related on the exchange sites. That exchange can be pretty fast. On the other hand, if it's the nutrients are literally part of organic matter that has to decay before it's released, that is going to be rather slow. And then if we start talking about dissolution of primary minerals, of the rocks themselves, that can in some cases be very fast, but in some cases can be very slow. Okay? All of those are going to have controls on this flux into this nutrient solution. Right? Which then means we have to actually think about what's going on here and here. We got plant uptake, we have exchange reactions. Those plants take that stuff up and that gets harvested. Potentially, stuff that's in the solution can be leaching. So what is the only other source for these nutrients to move into the system? Basically, us bringing in amendments. If we're harvesting the nutrients, not just the plant. The result of this is basically twofold. One is the amount of nutrients that are in the soil solution depends upon the history of that site. Okay? The amount of nutrients that are in this pool right here are going to depend on the history of that, the history of that, the history of that, and the history of this. Does that make sense to everybody? Okay. Now, the other interesting thing about this, the other result of this, is that you have to consider that each one of these nutrients are going to behave differently in that soil solution. Some of these are more soluble than others. Some of these are going to be coming in as fertilizers, some of them are not. Some of them are preferentially going to be leached from the system. Some of them are going to be preferentially lost from harvesting. Okay? Because of this history, you have to think about how these nutrients are individually going to behave as well. And this brings us to this idea of the tank model. Okay? We've looked at this before, okay? But just to remind ourselves, basically what we're looking at here is this tank represents all of the nutrients that are in the soil. This little spot right here is basically the nutrients that are in this soil solution. This is basically the reserve, primarily the cation exchange capacity, the stuff that's in the cation exchange capacity. Though it also could be considered, in a sense, the stuff that's in the structure of the organic matter. Okay? Now, as this nutrient pool is dropped down, the fertility of the pool is dropped down because of use, you're going to have an equilibrium between the active pool and this reserve zone, the exchangeable area. Okay? Now, what we would do is we would add fertilizer, and in the process of adding fertilizer, we would raise the nutrient levels in both the active solution as well as on the cation exchange sites, the reserve areas. Does that make sense? Okay. Now, let's think about what that actually means for the plant. Okay? Here, we've brought up the fertilizer all the way up to here. Okay? Optimum fertilizer levels, optimum nutrient levels in the soil. Okay? So here we start. The plant starts sucking nutrients out. What's going to happen to that active pool? It's going to drop down, but then it is going to equilibrate with the reserve area. It's going to drop that pool down a little bit and raise the active pool right back up. Not quite as high as it was in the beginning, but it's going to raise it back up. Okay? Now I keep drawing off of this, and I'm going to keep drawing this down, and I'm going to keep getting equilibration between the two of them. Right? Yes? No? Okay. Yes? Okay. Now, the question then is, what is the nature of this pool? Because this is really what we're managing. The plants are managing this for us. We're managing this. We're managing the whole system. So let's look at that tank analogy again, but let's look at it from a perspective of two different pools. Okay? Here is a pool with a low CEC. Here's a pool with a high CEC. Both of them, because of their drop in fertility, right now they're both pretty infertile. Does that make sense? Now, if I want to bring their fertility back up, I want to add fertilizer in some form. Now, to do that, because the capacity here is so small and this is so large, the amount of, let's imagine it's phosphorus, the amount of phosphorus that I would have to add as fertilizer to this system, to these two different soils, if I add X amount right here, it is going to raise the amount of phosphorus very much higher in this system than in this system. This line represents the low capacity soil. This line represents the high capacity soil. Does that make sense to everybody? If I am adding the same amount of fertilizer, here, no, stop. Let's turn it around. If I have the same amount equilibration rate in both soils, okay, starting, let's actually go back one more. Okay, I start at a high fertility soil, right? I put the same kind of plant in. They both have equal amount of demand. Now, if I have two soils, one soil with a large pool and one soil with a low pool, and they both start at the same amount of nutrient availability. There's the same level. They're all at this level, right? All the way across the board. If I put the same kind of plant in both of these, what's going to happen to the nutrient pools? In both of them, it's going to drop, right? It's initially going to drop down here, down to here, down to here, okay? But to make this back up, because there's going to be an equilibrium, this is going to come back to here. This line is going to drop here. This is going to come back to here. It's only going to drop here. So this guy now has availability to here, but this guy has availability to here. Does that make sense? Now, flip it over, though. Think about these when they're at their lowest point. Actually, I don't have to do that. We got that as a slide. Here they are at their lowest points. Which one of these is going to take more fertilizer to bring it back up to the high level? The high capacity one, because I put in the same amount in each case. Say X amount, 100 pounds. In this one, it's going to bring it up this far. In this one, it's going to bring it up a lot higher. Does that make sense to everybody? Yes? Okay, question.
How quickly does it reach equilibrium, especially compared relative to the rates that plants are taking it up?
Okay, so the question is, how quickly is it going to reach equilibrium? How quickly, if I draw this down, how quickly is it going to recharge in essence? Okay, that level, that recharge is going to be based on these three things. If it's a cation exchange reserve, it's going to be fairly fast. If it's organic matter decay, it's going to be fairly slow. If it's disillusionment, it's going to be slow or fast depending on what's being dissolved. Okay, does that make sense to everybody? So the question was, if we get to this state, or I should say, if we get to this state, how long is it going to take to get to that state? How long is it going to take to get back there? And it's going to depend upon this reserve pool. If it's cation exchange capacity, it's going to be fairly quick. If it's organic matter decay, if this pool here is basically an organic matter, it's going to be slow. Now, a good example of this is Dillman Hill. You guys, at this point, we got the results back from Dillman Hill last week, right? Everybody had really high levels of nutrients. Yet we still suggested giving fertilizer to those plants. Why? We had high levels of nutrients, yet we still asked. Why?
The high levels of nutrients could be in solid forms of some kind, like organic matter. So adding compost makes nutrients more available, or the nutrients in the compost are more available.
At Dillman, your answer is right. At Dillman, what are they using for fertilizer? They're adding it every year. It's compost, okay? Which means a lot of those nutrients are tied up in that organic matter. And if it's tied up in that organic matter, it's going to be the slow decay. So this return is going to be a lot slower. And that is why we suggest adding a little fertilizer for certain stages of those plants' life. Go.
So in that scenario, though, if you're consistently adding compost, would you theoretically be at a steady state where you would have to add fertilizer because you had compost there?
And the answer to that is yes. So the question is, in Dillman's case, if we're consistently adding organic matter year after year after year, would you still have to, because this stuff is decaying, okay? So supposedly it should, at some time, it's got to become available. In Dillman's case, because they've been adding this organic matter year after year after year after year, they don't actually have to follow the recommendations because there is, in fact, a lot of nutrients there that are, in fact, available. They may not be as available to certain stages of the plant's life as they're needed, but this is actually a deficit of the nutrient analysis of the protocols that we do for a recommendation. We're basing our recommendations, we're doing chemical analysis, we're basing our recommendations on sort of history of the site. Part of that history of that site that we don't take into account is this continual amendment of organic matter. Okay? Some of you guys had 17 to 25% organic matter in those beds. That's not from a traditional agricultural process or practice, whether it's a horticulture operation or field crops. Those are levels of organic matter that are usually not found. You're lucky if you find 7, 5, 8, 7%. Okay, you are really, really lucky. And in this case, we got 25% organic matter. That's well beyond what we usually see. The reason being, they add this stuff every year. Okay? Questions?
This has been a production of Cornell University, on the web at cornell.edu.